me see. Okay, so chapter 4. Chapter 3 gave us an overview of the prokaryotic cell. Chapter 4 is going to go over what the cell needs in order to survive. What are the factors that affect cell growth? And first, we look at cell growth itself. And when we look at cell growth, we're looking at two things. We're looking at individual cells and how they grow, how they divide, and then we look at the population growth. So obviously, one depends on the other. When an individual cell divides, bacterial cells divide by binary fission, and binary fission begins by the cell accumulating nutrients and dividing its DNA, enlarging, and then eventually dividing in half to create two cells. So we have cell growth first, and then cell division. And from one cell, at the point of cell division, we're going to have two cells. So we'll go from one cell to two cells. Then when each one of those cells divide, goes through binary fission, the same process will repeat, and we're going to end up with 4 cells. So we went from 1 to 2 to 4. When each of those 4 cells divide, we're going to have 8 cells, and then from there, 16 cells, and you get the picture. So this is one of the characteristics of population growth in bacteria. Because bacteria divide by binary fission, they double in numbers every round of division, and they grow in the population grow in numbers very quickly. Each division is called a generation. So going from 1 cell to 2 is 1 generation. From 2 to 4 will be the 2nd generation in this example. From 4 to 8 is the 3rd generation in this example. From 8 to 16 is the 4th generation in this example. So this population that we have here in this picture has gone through 4 generations. Now, the time that it takes for a population of bacterial cells to go through 1 generation is the generation time, or G. So in this case, you can see that the generation time is 30 minutes for the 1st generation, another 30 minutes for the 2nd generation, another 30 minutes for the 3rd, another 30 minutes for the 4th. So providing nothing changes in the population, the pH, the temperature, the level of nutrients, provided all of that stays the same, the generation time remains constant in the population. In this example, the generation time is 30 minutes per generation. Every 30 minutes, the population doubles in numbers. So you have to become familiar with those concepts and definitely know what it is, generation time. Generation time is the time that it takes for a population to go through 1 generation or doubling. We typically measure generation time in hours. Okay. And you should be able to tackle simple problems pertaining to generation time. Generation time varies depending on microorganism, depending on conditions. So, for example, Mycobacterium species have very long generation times because of their waxy cell wall. Generation time for most Mycobacterium species is 24 hours per generation, whereas the generation time of a more common bacteria, like E. coli, is 20 minutes per generation under optimum conditions. Again, this is important because the minute you change something in the environment, that is going to affect how fast the population grows, or the generation time. So the generation time of a population is definitely dependent on the environment and the availability of nutrients. So this is just to illustrate exponential growth, which is what bacteria experience, bacterial population, versus arithmetic growth. Arithmetic growth proceeds 1, 2, 3, 4, 5, 6, 7, 8. Exponential growth doubles every set length of time. So it would proceed from 20 to 40 to 80 to 160 to 320 to 640. So within a matter of a very short time period, there will be lots of increase in population. So there is a very dramatic difference between generation time exponential growth and arithmetic growth. So an example here is, give me just a second, maybe, okay, give me a second. Let me respond to some of the other students in the group that are not being able to get in. Okay, use the lab link. Let me make sure the lab link is working. Give me just a second, guys. Okay, let me check on their link. Okay, let me check on their link really quick. So let's just put this on hold. I'm going to, yeah, let me check on the link real quick. I'm not going to stop the recording, otherwise it's going to be very small recording. Give me just a second. I'm going to check on their link. I'm certain that I have put a link in there. Yeah, there's a Monday session link. Oh, I know what it is. Maybe they're trying to get into the. Okay, yeah, the link is working. All right. Let me send a message. Okay, very good. Yeah, I think that was the problem. All right, so moving on. Oh yeah, I'm sorry. Yes, this example right here just illustrates how quickly bacterial population may grow in numbers. You have 10 cells. The cells are going to go through doubling in numbers every 20 minutes for a period of 4 hours. So every 20 minutes, the population doubles in numbers, and we start out with 10 cells. So that means that in 1 hour, we're going to have 3 generations in 1 hour. But we're going to be doing this for 4 hours, which means that the population is going to double in numbers 12 times in 4 hours. We start with 10 cells, which means that we're going to go from 10 to 20 to 40, 80, 160, 320, 640, 1,280. So that would be 1, 2, 3, 4, 5, 6, 7, 8. What is that? I mean, my math breaks down now. That's 1,280. So that would be 6, 5, I think 2,560 at the end of 8 generations. And then we carry that all the way to 12 generations. We end up with 40,000 cells, which is a tremendous number in a matter of 4 hours, starting out with 10. So that gives you an idea of how quickly the population of bacteria can grow. And that's because they have very short generation times. That's essentially why, because they can double in numbers very, very quickly. So again, be able to solve simple mathematical problems using the formula G, generation time, equals T, which is the incubation time, over number of generations, which is N. So in this example, they're giving you G, and they're giving you T. They want to know N. So it's a matter of solving for N and then doing the math. So T is 4 hours times 0.33 hours per, make sure I'm doing something. Give me just a second. Oh, yeah, my math is wrong. Give me just a second. Let me clean this out really quick. Okay, so G equals T over N. I want to solve for N, which means that we're going to have T over G. That will make more sense. So N equals 4 hours. And just so that you know, I caught my mistake because of units. Because the way I had set it up, I was not going to end up with generations. And I realized immediately I did something wrong. So follow the units, and that will help you. If you're catch mistakes, hours per generation. So if I'm after n, just a second, there we go. How many generations? I'm after n. I should end up with units of generation only. Okay, so this is my setup. Hours eliminate. All I have left is generations, which is what I should have left. So n equals 4 divided by 3, which should be about 12, I think. 12 generations. This is another one. They're giving you n, they're giving you t. Now they want you to come up with g. Easy enough. T over n. T is 6 hours over 10 generations, which that gives us 0.6 hours per generation, or a little over half an hour per generation. So you should be able to come up with or solve these simple types of problems. Any questions over these? Okay. Also, keep in mind, generation time is the time that it takes for the population to double in numbers. So in this question, which organism grows faster, one with 0.5 hours per generation or one with 0.35 hours per generation? Well, this one is dividing over, it has a shorter generation time. Therefore, it's growing faster. In 1 hour, this one will double in numbers 3 times. This one will double in number 2 times. So keep in mind what it is that we're measuring with generation time. Okay. We have talked about biofilms several times already. Just review biofilms, what they are, what the importance is. And one thing about biofilms, which the book talks about, and I don't really emphasize, I probably one of these days I will emphasize it a little bit more, but one of the importances of biofilms is that this is a natural environment for microorganisms. You know, that gunk that appears in pipes, that's a biofilm. That stuff that sometimes appears in our commodes, that's a biofilm. A plaque in our teeth, that's a biofilm. And the importance of biofilms is that this is how organisms live together in their natural environment. And turns out when organisms are found in their natural environment with different types of bacteria growing together, they behave differently than when they are isolated in the laboratory. And that's something that we're beginning to realize. It's called quorum sensing. They have a sense of their numbers. They have a sense of who's around. They communicate with each other through chemicals, and they will change each other's behavior. And they may do things in a community of cells that they will not do when they're isolated in a pure culture. And that is something that we are exploring and beginning to understand. Importance of biofilms. Biofilms appear in infections, making it a lot harder for antibiotics to reach and kill bacteria and infections. Obviously, they also have a commercial importance. They also can clog up medical devices, which again makes them medically important. Okay. Now, pure cultures, we talked about pure cultures in the lab. We talked about aseptic techniques. So review what a pure culture is, what are aseptic techniques. Remember, a CFU is a colony forming unit and represents one cell, which will be the progenitor of a colony. Know what a medium is, what a culture. A lot of this is we get it from lab. So that's why I'm kind of going through it very quickly. Okay, agar, we talked about it in lab, is the substance that is used to make a medium solid. It's similar to gelatin, but it has some important differences. First of all, it cannot be destroyed by high temperatures. So we can sterilize it, and it will, after running through the autoclave, it can still work. It stays solid for a higher temperature, 45 degrees centigrade, and actually higher than that, usually 50, 55 degrees centigrade, it remains somewhat solid. Gelatin becomes liquid at 37 degrees centigrade. So we could not use it in medium that is going to be incubated at 37, which most of the time we incubate at 37 because that's body temperature. More importantly, it cannot be degraded by most bacteria, whereas gelatin can. So know the advantages of agar over gelatin. Okay, now this is an important principle. How a population of bacteria grow. And there are two ways of growing them. One is a closed batch culture. Another one is an open batch culture. A closed batch culture is what we do when we grow organisms in a test tube or a plate. And it's called closed batch because whatever is there is all the population is going to get. We're not going to add new nutrients. We're not going to remove waste. Therefore, whatever happens to that population over a period of time, it would be the growth of the population and then the death of the population. So in other words, this is not a system that allows for continuous growth of a microorganism. Eventually, everybody's going to die because it's a closed batch culture. We're not adding new nutrients, we're not removing the waste. And the whole population is not going to remain steady, it's going to change as the nutrients deplenish and the waste accumulates. So this is not a long-term method of growing bacteria. However, it's very inexpensive, and most of the time, for the purpose, it works just fine. Contrast a closed batch system with an open system. In an open system, you're going to have the population of microorganisms growing in an environment in which new nutrients are added regularly and waste product is removed regularly. So the population can remain indefinitely at a steady growth. Open batch systems require special equipment, and this is what the industries that grow microorganisms will have. Will have these open batch systems that allows them to have continuous grow microorganisms producing whatever product they want the microorganism to produce. It will be alcohols or amino acids or acetone, whatever it is. In a lab, however, in a medical research lab, a closed batch teaching lab, a closed batch system is all we need. So when you're growing bacteria in a closed batch system, the organisms are going to go through a series of phases of growth. Remember that what we're going to do, and you did some of these in lab 3, is we have the culture of the organism, and when we initiate a new culture, we transfer some of the organisms from one culture to another. So the new organisms now, in the new culture, first thing they have to do is get used to the new environment. That happens in the lag phase. They begin to accumulate resources and begin to replicate DNA, and then slowly, as they get out of the lag phase where there is no growth, they're just getting ready for growth, but there's no growth yet. The numbers remain steady initially, and then slowly the population begins to double in numbers, and they go into an exponential or log phase, in which the population is going to double in numbers every generation time. They're going to grow very quickly, and as that phase continues and nutrients are depleted and waste begins to accumulate, the population will slow down and will enter stationary phase, where the growth is leveled off. As nutrients continue to deplenish, waste continues to accumulate. Eventually, the number of bacteria dying is greater than that of the bacteria dividing, and the population numbers begin to decline until they go into the prolonged decline. So the growth curve in a closed batch system is something like this, where you have the lag phase, the log or exponential phase, the stationary phase, death phase, and then phase of prolonged decline. In theory, there's never complete death of the entire population because in theory, there will always be mutants that are able to survive in the environment of the culture. In real life, we never get to that point because we subculture again and get a new culture. So you should be able to identify the phases of growth in a closed batch system. You should know the lag phase is the initial phase where there is no change in numbers, so population remains steady. And as the population begins to multiply, it's going to begin to double in numbers and grow very, very quickly. That's the log or exponential phase. This is the best time to identify organisms, to make gram stains, to calculate generation time. This is the time when organisms are most sensitive to antibiotics because that's when they're actively dividing. And most antibiotics work as the cells actively divide. So if the cell is doing nothing, like in the lag phase, an antibiotic is not going to work. It has to be an actively dividing cell for most antibiotics to work. Now, this exponential log phase, it is a major part of the cycle, and it does have phases. So you do have, the beginning of the phase, which is different from the later stages as we get closer to the stationary phase. So at the very beginning, or when the middle of beginning of this phase, when the population is very strongly dividing, it is making products of metabolism, which we call primary metabolites. And those are going to be things like alcohols, acids, amino acids. So this is the stage at which an open batch system will seek to permanently keep their cultures, because that's when the bacteria is making stuff. Actually, secondary metabolites can also be very helpful. As the log phase begins to end, so this is towards end of log phase, the cells begin to change and begin to get ready to go into the stationary phase. In other words, at that point, there will be less nutrients, more waste, and the bacteria will begin to secrete things like antibiotics to kill their competition. Bactericidins, which are again products that kill other bacteria. So those are the secondary metabolites, which are produced towards the end of the log phase. So know what a secondary versus a primary metabolite are and when they're produced. Stationary phase, by definition, is the time when the number of cells dividing equals the number of cells dying. And then the death phase is when the number of cells dying is greater than the number of cells dividing. So again, know the phases of growth. One thing you're going to be doing in lab, not this next time, but the next one, is trying to create a growth curve like this one right here. Okay. Any questions? All right. Okay, so now we're going to look at the factors that influence bacterial growth. And these are the list of the factors that can influence how well or how poorly a population grows. Temperature is one of the first factors. Temperatures is linked to the work of enzymes. So most enzymes work best at a range of temperatures. And typically there is a very narrow range in which they work at their very, very, very best. So that's what we call the optimum temperature. So at the optimum temperature, the G is very small because the population is dividing at its quickest. This is typically closer to the upper range. So let me explain these. Actually, let me show you these. Okay, so this is the growth curve of an organism. The peak is the optimum temperature. That's the temperature at which the organism grows its fastest. G is at its smallest. That's the best temperature for growth. So in this organism is about 95 degrees centigrade for this particular organism. However, the organism could grow at temperatures as low as about 75 and as high as about what, 105 or something like that? No, 115, more or less. So this is the minimum and maximum range of temperatures at which the organism grows. Notice that 95 degrees centigrade is closer to the upper range than to the lower range. So a growth curve is typically going to be lopsided in that the peak of the curve is going to be closer to the upper range of growth. The reason why is because once we begin to move at temperatures higher than the optimum, very quickly the high temperatures kill the enzymes and kill the population. Whereas low temperatures are not going to kill the enzymes, are just going to slow them down. And so organisms typically have a larger range of growth at lower temperatures from the optimum than high temperatures from the optimum. Okay, so that's an important concept when it comes to temperature. High temperatures are more damaging than low temperatures to an organism. Now, aside from that, we can classify bacteria according to the temperatures at which they grow. So sacrophiles are bacteria that like cold temperatures, and they can grow at temperatures as low as minus 5 degrees centigrade. 0 degrees is ice, so this is below freezing, and as high as 15 degrees centigrade. These are the bacteria that are found in very cold regions of the earth. Cyclotrophs, on the other hand, are kind of cold-loving, but not as dramatic as the sacrophiles. Cyclotrophs have a range of 0 degrees to 30 degrees centigrade. As a point of reference, room temperature is taken as 25 degrees centigrade, body temperature is 37 degrees centigrade. So these guys like something cooler than body temperature, a little bit warmer than room temperature. However, they can grow at low temperatures, like the refrigerator. 4 degrees, 6 degrees, 9 degrees centigrade is refrigerator temperatures. So they can grow very easily in the refrigerator. These are organisms that are important when it comes to food spoilage. Now, the ones that are really important for us are the mesophiles, because these are the ones that can grow very well at 37 degrees centigrade. And actually, a mesophile, most of them have an optimum growth temperature of 37 degrees. So the range is room temperature to a little higher than body temperature. 45 degrees centigrade is a very high fever. These are pathogenic organisms. The optimum temperature ranges between 35 and 37 degrees centigrade. So that's why they are all pathogenic and opportunistic pathogen are mesophiles. Now, keep in mind microorganisms cannot read, so they don't know what their range of temperature is supposed to be. And when we do in the lab experiments and try to figure out the range of temperatures at which some of these organisms grow, we find some mesophiles can grow as low as 10 degrees centigrade. Okay, some can grow as high as 50 degrees centigrade. So this is an average for most, but keep in mind That's not set for every single mesophile. The key, however, between a sacrotroph and a mesophile is the optimum temperature. A sacrotroph has an optimum temperature of about 25 degrees, maybe 30 or 28, close to 30. Okay. A mesophile has an optimum temperature between 35, 37 degrees centigrade, sometimes maybe even 40, but it's higher than the sacrotroph. A sacrotroph doesn't grow well at body temperature. A mesophile does. On the other range of the scale are the thermophiles that need high temperatures, between 45 to 70, with an optimum between what? This is the thermophile. Actually, that's about, according to these, the thermophile grows more closer to 85. Here it says 70. I took this from the book, in this picture. I don't know where I got this. Oh, it's actually from a Grow Hill book too. But anyway, so these pictures telling us more like 85, not 70. The point is, optimum will be around 70, 75, maybe 60, or thermophiles in our lab, the optimum is about 55 to 60. That's the optimum for the thermophiles we grow in the lab. So these are organisms that can grow in hot springs. And by the way, thermophiles can grow in our hot water heaters. Thermophiles are some of the organisms involved in yogurt making, for example, too. So a lot of them are useful as far as thermophiles. Hyperthermophiles, those are the extreme thermophiles. They grow at incredibly high temperatures, 70 to 110 degrees centigrade. Keep in mind, 100 degrees centigrade is boiling water. So the optimum for these guys is going to be above boiling water. These are members of the Archaea domain, and they're found in very limited and extreme environments. Having said that, hyperthermophiles, because they can function at very high temperatures, are used in research, like some of the molecular biology procedures like PCR rely on hyperthermophiles. So they're useful for other purposes. So definitely know the different kinds of organisms, given a range of growth, of temperature growth, be able to put an organism into one of these categories, give it an optimum temperature, know what the organism is probably, which was the category of the organism, etc. Yeah, as far as pathogenic and opportunistic pathogens, pretty much all of them are mesophiles. A few of them may be a little bit into the cyclotroph side because they may grow only in parts of the body where their temperature is lower. But for the most part, you can throw a blanket and say they're all mesophiles. This is another important concept which I've already explained, and this is just to emphasize, make sure you understand the temperature growth curve. The peak is the optimum temperature. At this point, G is at its shortest. Notice that the peak is lopsided. The minimum growth temperature is broader than the maximum growth temperature. You should know that the reason why is because high temperatures destroy enzymes, whereas so the nature's enzymes versus low temperatures, it slows them down. The minute you raise the temperature again, they'll come back to being active. So if you want to kill an organism, don't give it a low temperature, give it a high temperature past the optimum. If you want to slow it down, put it in the refrigerator, give it a low temperature. If it is a mesophile, obviously. Okay, so you will do an exercise in which you'll explore ranges of growth of microorganisms by temperature. Okay, now the next parameter that can affect bacterial growth is atmosphere oxygen, and this one is very important. Okay, so first of all, why is oxygen so important? And that goes back to making energy, ATP. And cells make energy, if you remember biology 1406, you were taught that they made energy by a process called aerobic respiration, which took place in an electron transport chain. And in the electron transport chain, oxygen was the final electron acceptor. We're going to review all these processes in chapter 6. But for now, oxygen is needed for the ETC to keep going and to be able to make tons of ATP. So aerobic respiration is a very efficient way of making energy, but it requires oxygen. The problem with oxygen, which makes us incredibly efficient, however, oxygen is ultimately a poison because it is an oxidizer. And in biology, what being an oxidizer means is it robs hydrogens from molecules. And living things are loaded with hydrogens. So oxygen is going to go around oxidizing our proteins and our carbohydrates and our lipids and causing toxic damage. So we are in a quandary. We need oxygen to make ATP efficiently, but at the same time, oxygen is killing us. So the solution was to create enzymes, to evolve enzymes that would take care of the poisonous forms of oxygen, like superoxides and hydrogen peroxides. The enzymes are called superoxide dismutases and catalases. So all living things that live in an oxygen environment and or use oxygen need these enzymes in order to survive. Okay, so that's the premise. That's why oxygen is important. When it comes to eukaryotic organisms that we are, well, most eukaryotic organisms are going to need oxygen. We're make ATP by aerobic respiration. That's not the case with prokaryotes. Prokaryotes have a much more diverse relationship with oxygen. So there are many different types of oxygen requirements for microorganisms, for prokaryotes. So let's go over those. And let's start with one that we're familiar with, the ones that need oxygen. So microorganisms that need oxygen are called obligate or strict aerobes. They do have superoxide dismutases and catalases. Makes sense. They live with oxygen. They use oxygen to make ATP. They have an aerobic respiration pathway, just like we do. Example, bacteria in the genus Micrococcus are strict aerobes. Then you skip to the other end of the spectrum, the obligate anaerobes. These are organisms that are killed by oxygen. They do not have SODs, they do not have catalases, they do not use oxygen to make ATP. They make ATP by anaerobic respiration and or fermentation. Again, these organisms are typically killed in oxygen. So, examples, members of the genus Bacteroides and Clostridium. So when you grow these organisms in a tube, for example, they will grow at the bottom of the tube, away from oxygen. When you grow the strict aerobes in a tube, and what we call a soft agar, or a thioglycolate fluid, they're going to grow on the top. Okay, so those are the two extremes. Let's go for the middle ground. These guys are incredibly lucky. These are the ones that can do everything. They can live with or without oxygen. However, they prefer oxygen because oxygen makes them more efficient. They have catalases and SODs. When they don't have oxygen, they have to become less efficient, making ATP through anaerobic respiration and or fermentation. When you grow them in a tube, they're going to grow all over with more growth on top because they're more efficient with oxygen. Examples, members of the species in the Staphylococcus genus, all of the genera in the Enterobacteriaceae family, for example, E. coli, members of the Enterobacter genus, Proteus, So these are genera in the family. Okay, Salmonella, Shigella. Anyway, this is a humongous family. All of them are facultative anaerobes. So we have strict aerobes, strict anaerobes, facultative anaerobes. Facultative anaerobes are the lucky ones. They can do everything. These are the unlucky ones, the aerotolerants. Okay, aerotolerants have no catalases. They only have SODs. Okay? The only means for making oxygen is fermentation. Fermentation is incredibly inefficient. They make very little ATP, which means they grow slowly. They don't like oxygen. They will grow in it, but they're not happy. They don't like it. In a tube, they're the tricky ones. They will grow all over. They tend to avoid the top, so there may be a little bit of growth on the top, but compared to the facultatives, which grow tremendously on top, these guys don't grow very well on top. All of the species in the Streptococci genus are aerotolerants. The last group are the microphiles. Micro means small, so they need a little bit of air, but not too much. Atmospheric oxygen has about 20% oxygen. These guys need between 2 and 10%, so about half to a tenth of what it is in atmospheric environment. They do need a high concentration of CO2, which makes them capnophiles. Capnophiles means CO2 lovers. The reason why they need less oxygen is because they do have SODs, they do have catalases, but they have small amounts of these enzymes. They do use aerobic respiration, but they need less oxygen. Helicobacter species and Campylobacter species are macrophiles. And in the tube, they would grow somewhere in the middle. So, know all of these groups, which ones have which enzymes, where will they grow? So here is an obligate aerobe. This is facultative anaerobes. These are the obligate anaerobes. This is your aerotolerant, and notice the difference between aerotolerance and facultative anaerobes. These are the macroaerophiles. And there is a chart that summarizes all of this information for oxygen requirements. I'll be able to answer questions like these. Where would E. coli grow? Which of these is a capnophile? Which of these organisms lacks catalases, so catalase negative? Which of these tubes could be growing an organism called Clostridium perfringens? Which of these organisms can use fermentation to make ATP? Which of these organisms does not have SODs? So be able to answer questions like that about oxygen requirements. Okay, any questions? Okay, so moving on. pH requirement is pretty straightforward. Most bacteria like an optimum pH is about 7. Between 5 and 8 with the optimum of 7. So most organisms are neutrophiles by far. A few may lack or may need an acidic environment. A few may need an alkaline environment. Fungi are slight acidophiles, but for the most part, most microorganisms are going to be neutrophiles. Now, some neutrophiles have managed to live in acidic environments. Helicobacter pylori, which is a microaerophile, colonizes the stomach, and so is able to live in such an acidic environment because it can split urease and make lots, secretes urease. Ureases are alkaline, so the ureases buffer the acidity. They make tons of CO2. They increase the pH of the stomach. They make ammonia from splitting urea into CO2 and ammonia. So they buffer the acidity. That's essentially what they're doing. They're buffering the acidity. And that's how they can survive in the acidic environment. We use extreme pHs to preserve foods. So we use bacteria that makes tons of acids and that can tolerate acidic environments to create acidity that prevents other organisms from conquering or colonizing that area, that food. So lactobacillus is actually normal flora in our bodies and in other things like milk, and we have been using them for thousands of years as a preservative for foods, without even understanding that the acidity was being created by microorganisms. Okay, water availability is the availability, whether the medium has water. Obviously, living cells need water to live. And now, lack of water doesn't harm endospores because they're dormant. Cysts of protozoans, which again are dormant, they're not active, so they don't need water. Living actively metabolizing cells do need water. We mentioned in chapter 3 that a hypertonic environment is more damaging to a cell, to a prokaryote, because the cell wall protects them from hypotonic environments. So in a hypertonic environment, they're going to lose water, shrivel, and die. So we use hypertonic environments to discourage the growth of bacteria. Now, halophiles are rare organisms that require incredibly high salt concentrations to grow, incredibly high hypertonic environments. This is the bacteria growing in the Dead Sea, in Utah salt lakes. Halotolerants, on the other hand, can't withstand high salt environments. They don't like it, but they can withstand it. Okay, huge difference. Important, the genus is Staphylococcus, mesophile, facultative anaerobe, halotolerant. The fact that it's mesophile tells you immediately it's an opportunistic pathogen. And it can grow in high salt environments. May not be happy, but it can grow on it. So this is the organism that can grow on the skin, which is very salty because of sweat. It can grow on dry meats, hams, jellies. So it can't colonize foods that most organisms, other organisms cannot colonize. So we use salted fish, dry food, dry meats to lower the water activity, create hypertonic environments, and preserve the food. Pure water has a water activity of 1, so anything below 1 will be will have less water. So bacteria can withstand water activities of 0.91. Fungi can withstand much drier environments. Again, with Staphylococcus being exception in that it can withstand pretty high salt, pretty low water activity. Okay. Now, the last part of the chapter looks at other requirements for growth. And obviously carbon, being that we are organic living things that have to make organic compounds, carbon is an incredibly important element that we need to attain somehow. So there are 2 ways in which living things in this planet can obtain carbon. We can either eat the carbon from other organic compounds, or we can obtain the carbon from the environment by taking inorganic CO2 and making organic compounds from it. So here we use organic carbon to make our own organic compounds. That's a heterotroph. Autotrophs get the carbon from the atmosphere in an inorganic form and incorporate it and make it into the proteins and lipids and carbohydrates they need for their cells. That's called carbon fixation. So an autotroph versus a heterotroph, their difference is how they get their carbon source. Another some bacteria have needs for special vitamins and nutrients called growth factors, and those have to be provided in the medium. Those organisms are called fastidious or hard to grow. Organisms are going to need an energy source, and the energy source can be from chemicals, in which case the organism is called a chemotroph, or it can be from light, in which case it's called a phototroph. So in order to create nutritional requirements, we take the need for carbon and the need for energy and then put it together into nutritional requirements. So remember, the need for carbon gave us autotrophs versus heterotrophs. So we take the need for carbon and the need for energy, and we could create 4 different energy or metabolic requirements. So we can have the photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. All right, now eukaryotes only have 2 of these basic groups. The photoautotrophs and the chemoheterotrophs. Photoautotrophs are going to be the living things that use light as energy and fix CO2 for their source of carbon. Those will be algae, plants. I know plants are not microorganisms though. Those will be your photoautotrophs. Chemoheterotrophs are going to be the organisms that use chemicals as their source of energy and carbons. That will be animals and fungi and protozoans. Plants are the photoautotrophs. That's it. Okay. Now, bacteria. Bacteria I shouldn't say bacteria, I should say prokaryotes, bacteria and archaea, have 4 groups instead of 2. Some prokaryotes are photoautotrophs, just like plants. Some prokaryotes are chemoheterotrophs, just like animals. All bacteria that are chemoheterotrophs, or I should say, put it the other way around, all pathogenic and opportunistic pathogens are chemoheterotrophs. Okay? The other 2 are more specialized nutritional requirements, which are typically not medically important. The chemoautotrophs are bacteria that can use chemicals for energy, but they get their carbon from CO2. Okay? These are organisms that can, when it says chemicals for energy, the chemicals could be inorganic chemicals. So some of the members of this group are what we call rock eaters. They can get energy from inorganic compounds and get their carbons from CO2, so they have no need for organic or other living things for their existence. They can live without needing other living things, which is actually pretty cool. The fact that they can do that. They literally eat rocks. Those are the chemoautotrophs. These are organisms that sometimes are found in mines, essentially eating rocks. Photoheterotrophs, those will be photosynthetic. They're going to use light for their energy source. However, they're going to use chemicals for their carbon source. So they don't have a Calvin cycle They are photosynthetic, but they don't have a Calvin cycle. So these are our microorganisms, bacteria, prokaryotes that live in polluted lakes, where they can use the light that filters through as their source of energy, and they'll use the organic compounds, dirt and leaves and decaying matter, as their source of carbon. So again, those are specialized photosynthetic organisms. So these two are groups are unique to prokaryotes. Do know, however, all medically important organisms are chemoheterotrophs because that's what we are. All right, that's it for the chapter. One of the most important aspects of the chapter is oxygen requirements, temperature requirements, and generation time. So if nothing else, focus on those concepts for this chapter. Okay, any questions? All right, I will